The human brain, the most complex organ in the known universe, begins as a flat sheet of cells. And within just a few weeks, it folds, shapes, and transforms into the early blueprint of your entire nervous system. Today on Inside Human Biology, let's follow this journey step by step. Overview. In this video, we'll explore how the brain forms from week 2 to week 8 of embryonic life, from molecular signals to the very first neurons. Let's start at the beginning. Stage 1. Gastrulation and germ layers. Around day 14 after fertilization, the embryo undergoes a dramatic transformation called gastrulation. This event produces 3 primary layers: the ectoderm, mesoderm, and endoderm. Everything in the human body will come from these layers, and the brain comes specifically from the ectoderm. Stage 2. Neural induction, how the brain is ordered to form. Next, a special region called the primitive node releases powerful molecular signals. These signals, including noggin, chordin, and follistatin, block a protein called BMP. Under normal conditions, BMP would turn ectoderm into skin. But once BMP is blocked, the ectoderm switches identity and becomes neuroectoderm, the tissue that will form the entire brain and spinal cord. This is the very first step in creating the nervous system. Stage 3. From neural plate to neural tube, neurulation. The neuroectoderm thickens into a structure called the neural plate. The edges begin to rise, forming neural folds, and the center forms a neural groove. These folds gradually move toward each other and fuse, creating the neural tube, the earliest form of the central nervous system. Between day 22 and day 28, the tube closes completely. If the upper opening fails to close, it leads to anencephaly. If the lower part doesn't close, it causes spina bifida. This is why folic acid is so important early in pregnancy. Stage 4. Primary and secondary brain vesicles. The brain takes shape. Once the neural tube is formed, the head end begins to expand rapidly. 3 major bulges appear, called the primary brain vesicles: the forebrain, midbrain, and hindbrain. Over the next week, these 3 divide into 5 secondary vesicles: telencephalon, which will become the cerebral cortex; diencephalon, forming the thalamus and hypothalamus; mesencephalon, the midbrain; metencephalon, forming the pons and cerebellum; myelencephalon, which becomes the medulla. By week 6, the basic map of the brain is already in place. Stage 5. Molecular patterning. Who decides which part becomes what? Inside the neural tube, chemical gradients guide the brain's architecture. Signals like sonic hedgehog, SHH, rising from the notochord shape the ventral or motor regions. Meanwhile, WNT and BMP signals from above shape the dorsal or sensory regions. Other molecules such as FGF, retinoic acid, and genes like HOX, OTX2, and EMX2 create the front-to-back pattern of the brain. These signals act like a blueprint, telling each region exactly what to become. Stage 6. Neurogenesis and neuronal migration. Around week 6, the cells lining the neural tube begin rapidly dividing. These early stem cells, called neuroepithelial cells, produce the first neurons and later glial cells. New neurons migrate outward in a highly organized fashion, forming the layers of the cerebral cortex. This process follows an inside-out pattern. The deepest layers form first, and the outer layers form last. Stage 7. Axon guidance and synaptogenesis, building connections. As neurons settle into place, they begin extending axons, long fibers that search for their target partners. Their path is guided by signals like netrons that attract, slits and semaphorins that repel, and efferents that create boundaries. Once connections are made, synapses form. These synapses then undergo pruning. Weak connections are eliminated while strong ones are reinforced. This is how early neural circuits begin to take shape. Medical and developmental importance. These early weeks are some of the most critical in human development. A lack of folate, exposure to alcohol, certain medications, or infections during this window can disrupt brain formation. This is why early prenatal care plays such an important role in healthy neurodevelopment. Conclusion. So, the human brain begins as a simple sheet of cells, and through precise signals, folding, migration, and connection, it transforms into the foundation of the most complex organ on Earth. If you want more deep dives into human development and biology, make sure to subscribe to Inside Human Biology. Tell me in the comments which topic you want next: cortical development, spinal cord formation, or brain disorders. See you in the next video.